Welcome. Today's Fireside Chat is going to focus on the official journal of SITC, JITC, or the Journal for Immunotherapy of Cancer, as it enters its 10th year of publication this year. I am honored to have with me today the founding and current JITC Editor-in-Chief, Dr. Pedro Romero, JITC Deputy Editor-in-Chief, Dr. James Goley, past SITC President, Dr. Lisa Butterfield, and JITC Managing Editor, Andrea Kuntz. Dr. Romero, 10 years for JITC. What a great accomplishment. You've been at the helm the entire time. I'd like to start this fireside chat with you providing us a little glimpse into what was happening 10 years ago that got this journal going.
Thank you, Pat. Sorry for my voice. Well, it was a tremendously exciting year for cancer immunotherapy, that of 2013. Ipilimumab had been FDA approved for the treatment of advanced metastatic melanoma after the demonstration that blocking CTLA-4 in those patients could extend survival, a first in melanoma since at least 30 years. Steve Hoddy, one of the leaders in the pivotal clinical trial, was our founding section editor for the clinical and translational tumor immunology section. Susan Topalian and the crew from Hopkins had shown in a landmark large phase 1 trial that PD-1 blockade was effective in treating lung and renal cell carcinomas as well as melanoma. SITC's leadership had decided to go ahead with publishing its own journal and trusted me with leading the bunch of editors that enthusiastically took on the challenge to edit the Journal for Immunotherapy of Cancer, exactly the year when science identified cancer immunotherapy as the breakthrough of the year, and also the year when our friends and colleagues at the American Association for Cancer Research launched their own tumor immunology journal, Cancer Immunology Research, edited by Glenn Drainoff. Really exciting times and a unique window of opportunity to enter the scene of scientific publishing of the results in a field that was growing and keeps growing exponentially.
What was the vision of the journal? Also, do you remember the first issue that was published by the journal?
So the first batch of papers were online on May 29, 2013. Now, together with Franco Marincola and Tara Whittington, we wrote an editorial introducing the Society's new journal. It started with these lines: the field of cancer immunology and immunotherapy is experiencing a tremendous increase in excitement with the advances in immune checkpoint blockade therapy. And we set out the tone to provide a full open access publishing of high quality research and literature reviews.
Hey, Pedro, you know, the journal has just had exponential growth. It's been really exciting for everyone in SITC and to see how prominent it's become so quickly, and you're really at the heart of everything. In fact, SITC's made a Pedro Romero Award that they're giving out every year for service to JITC. What's it feel like, Pedro, to have an award named after you? Are your kids proud? Tell me about that.
It was a tremendous honor, quite unexpected, and I feel humbled by that distinction. We all really did was to really work hard for achieving our objectives, and is very rewarding to be recognized in that way as well.
Dr. Butterfield, you were SITC president when the journal just really started to take off and get its first impact factor. You really helped it become a global journal. Can you tell us about that?
Sure. Thank you, Patrick. This is fun to think back 10 years to those days when, as a society, we were growing so fast, and we really had, we had a to-do list. We wanted to be able to have our different committees have a voice where they would be heard, where different guidelines could be published, the voice of the society. And so it was on the to-do list of the executive committee for many years, and we talked about it, and we dabbled a bit. We had sort of an experimental transitional period where we thought about how we could do this, and it was a big decision to really jump in and go ahead and create our own open access journal and to decide what its structure would be. And so it was, but we just kept circling back to the needs of the society and our members. We needed to have open access, and for a lot of us in the field, we knew that things like human biomarker research or really interesting case reports where you dig in, those could be very hard to publish, but we knew that they were interesting and important for the field. So to have an opportunity to have what SITC knew was important for our members and for the society, to have that voice, that was, I think, what pushed us over to really take this on and learn so much more about journals than I ever thought I would learn, as a research scientist.
Absolutely. I remember as long as I've been involved with SITC, we've been talking about having a journal, but I'm just so happy that all of you on this screen helped to execute and make it a reality. It's just been so much fun. And one of those really important people are Andrea Kuntz, the managing editor. Andrea, you are keeper of the metrics too. Tell us about the impact factor and what a difference that made, and about other metrics of the journal.
Yeah, no, absolutely. Thanks. I'm really glad to be here to chat about JITC. I think we all have such a love for it. Kind of thinking back even to what Dr. Butterfield mentioned, when we first got our initial impact factor in the summer of 2018, it was a big ordeal for the journal. We had we had about 15 submissions per month. And then overnight in July, the next month after we had it, we jumped up to 75 submissions. And that was, again, just overnight. So really the impact of that, the effects of having an impact factor really opened the journal up to much larger audiences and a broader network of researchers. And we felt it throughout. I mentioned the impact factor because it is just so well known throughout the community, throughout publishing, and very clearly, it's something that entire countries and institutions require for their researchers if they're going to publish somewhere or of a certain level. So it has the greatest potential to create an increase in submissions and publications and awareness. So that's why we tend to mention it more. There are others as well, our SJR scores and CITE scores, all where the journal really stands in this great reputable position. But again, it's just thinking back to with that impact factor, having the editors in place that we had was extremely helpful because, they went from doing business a certain way and not having to be called on all too often for a few submissions to suddenly having to be all hands on deck. And, thankfully, we had a few months to kind of prepare and meet with our editors, let them know about what's going to be coming. We got a chance to even update our website and make sure it's user-friendly for the new traffic that would be coming. And it really helped having those people in place and ready to go to take it on. And so it's, by all means, everything we had to juggle with that was a huge success due to the editors that we had in place.
It's wonderful, Andrea. Why don't you let us know the most recent impact factor?
Yes, so our current impact factor is 13.751.
Yeah, that's just a wonderful accomplishment in such a short period of time. James, thanks so much for joining us, and a couple congratulations for you. One, you were the first recipient of the Pedro Romero JITC Service Award. You got that in 2021, so congratulations on that. And also congratulations on recently your appointment to be deputy editor of JITC. So congratulations, both well-deserved awards.
Well, thank you so much. It's been fun. Before I became the deputy editor-in-chief, I was the section editor of the clinical translational section. And, when I started in 2017, Patrick, it was amazing. We were getting about 4 manuscripts a month, but when 2018, when the impact factor, as Andrea mentioned, came out at 8.4, we mushroomed a 5-fold increase in number of submissions to that section. And by the next year, I asked for additional help. So there was an additional section editor added. We also split off 2 sections there, the oncolytic and local therapy section and the cell therapy and immune engineering section. And that section, the clinical translational section, has continued to grow every year. So in fact, last year, there was almost 700 submissions for that section, which is about 2 papers a day. And our acceptance rate has gone down from about 50% when I first joined to about 25%.
Wow. And translation is part of SITC's secret sauce, don't you think? There are a lot of really strong basic immunology journals, a lot of strong clinical, but SITC really goes from lab to clinic and back. And so tell us, do you think that was a unique section within JITC? And do we have a unique take because of the history of SITC?
Yeah, absolutely. So I think that there are a lot of, this really is the heart of what we do, right? And so this is perhaps why it is the section that has the most papers submitted to this. But I think that what we've seen over time actually is not just an increase in the number, but we've seen a change in kind of the type of manuscripts that we've seen. Also, we've seen more clinical trials, including randomized clinical trials, more combination studies, and more international submissions also.
Wow. Well, real exciting. Another really important area and a secret sauce of SITC is biomarkers. So Dr. Butterfield, you were head of the biomarkers section. Tell us about biomarkers, the involvement with SITC, how it's uniquely poised in JITC, and where you think the future of biomarkers for immunotherapy lies.
Oh, thank you, Patrick. It was not assumed from the start that we would have a biomarker section. That was something that we actually debated a little bit initially around the table. But SITC for sure has always had a very active biomarkers community and very active committees. And so, it seemed like a natural fit. And in the beginning, we had to push for submissions because the journal wasn't known. There was no impact factor. And a great idea that our colleague Nora Desis had was to have technology primers. And so, short reports featuring biomarker technologies. And so that was a great way to drive interest and to have new things that would appear in that section. And then all of the work of the biomarkers committees, committees and subcommittees on pathology and circulating biomarkers and about the regulatory compliance in biomarker work. So all of those things appeared in the journal and continue to be great reference materials for the field. So we had, I think, a strong start from the society interests. But it's been a lot of fun to watch how it's evolved. And so, as I look every month at the updates and the featured articles, some of the things I've noticed lately are circulating biomarkers of shed co-stimulatory and checkpoint molecules, shed PD-L1, shed 4-1BB ligand, shed ICOS ligand. So, this is an area that I think has really gotten traction, and some of those early papers, did appear in JITC, and now there's validation in more and more clinical trials. So that's one of the areas that I think is a lot of fun. And these were papers that were hard to publish, but knowing that we have an audience, knowing that we've got a whole section dedicated to this, it's been fun to see that evolution. And now we're also seeing combinations. So not just a single biomarker in a clinical trial, but how tumor mutation burden and PD-L1 and gene expression profiles together can work as an important biomarker for tumors. So there's been some really great work that's now been coming out in that section.
Lisa, let's talk a little more about the liquid biomarkers and the circulating biomarkers. With targeted agents, it's pretty straightforward. You're looking for the mutations that are circulating around and you target those mutations. The challenge with immunotherapy is that there's an architecture that's going on in the tumor microenvironment, and knowing that architecture may be important for biomarkers. How is this solved for with circulating biomarkers, and where do you think the blood biomarkers, the liquid biomarkers, are going to hold in the future for biomarkers in general for immunotherapy?
Well, it continues to be the holy grail. We'd much rather just take a tube of blood than to have to really interrogate a tumor biopsy that's hard to collect. So the blood, has always been an area of interest and of investigation. And, and people who have thought that, it wasn't really even worth looking for biomarkers there. I think JITC continues to show month by month that there are important correlates in the blood. And as you say, Patrick, the looking just for that mutation in a circulating tumor DNA, is the tumor coming back? Is it not coming back? That's simple. And there's nothing simple about the immune system. So that's why, as I mentioned, the circulating checkpoints and costimulatory molecules, we didn't even know they were shed. And now we're trying to figure out whether they're in exosomes on the inside, on the outside, and are they just free proteins? And what difference does it make what the source of the circulating biomarker is, that subcellular localization or is free protein? So there's a lot more work to do always, but these, I think these molecules are now, really shedding mechanistic light on what's going on with our therapeutics targeting all of these important molecules.
Yeah, the exosomes really interesting. I think I just saw another paper in this month's JITC about exosomes and how they could cause immunosuppression. So that's another great example of circulating factors and biomarkers, as well as, I guess, tumor mutation burden, which you can always get from circulating tumor cells, right?
Yeah, there's also the time considerations. There's no lack of complexity to when you do that test relative to your therapy to look for an immediate response. It's really more of a PK, PD kind of thing versus the sign, the early warning that the tumor is going to go away or it's not. So I think that section is going to be around for a while.
That's great. Pedro, there's just so many great papers coming out of JITC these days. Can you tell us some of your favorites? Well, actually, you can't have favorites. You're the editor-in-chief. So tell us what some of the most prominent ones are and why.
Thanks, Patrick. We actually compiled the list of the 20 most cited papers of all time through 2021. And this group includes several reviews and position articles and guidelines. And in fact, the most cited paper of all time is the Managing Toxicities Associated with Immune Checkpoint Inhibitors Consensus Recommendations from the SITC Toxicity Management Working Group. It has gathered, up to now, it has surpassed 1,000 citations, and it is the first JITC paper to do so. So many of these top cited articles are also the highly accessed, but several were not. There are some that are highly accessed and yet not so frequently cited. That's in all a mix of different article types, including everything from reviews, position statements to a case report and a commentary.
Now I'm going to open up a question to the whole group. You see the cutting edge, you see all of the papers published and unpublished coming in. What are those areas of science that you think are that are coming along and really the hottest areas right now? In our SITC retreats, we've talked a lot about immune resistance, the tumor microenvironment, but what other areas are you seeing that are the most important to really take us to the next level in immunotherapy for patients?
So I think one area is synthetic biology and cell therapy. I think we're seeing a lot of interest in that area, and we're starting to see, can we get evidence of activity in solid tumors? I think also another area is bispecific antibodies, and not just necessarily T-cell engagers, but perhaps other things that are bringing cytokines or chemokines into the tumor microenvironment.
Maybe I could say the tumor microenvironment, as you mentioned, particularly from the perspective of using multi-omics approaches and trying to glean out the mechanisms underlying primary resistance, secondary resistance. Another area is, as Lisa just mentioned, that of composite biomarkers and liquid biopsies. And I think that is evolving tremendously, and probably is going to push the field big time forward.
I agree with all those. I'm very excited about synthetic biology in adoptive cell therapies and novel cell types. I wouldn't have thought an engineered macrophage, would do anything for more than 5 minutes in a patient. And we've seen exciting data from those, and, gamma delta T cells. So that's, I think that area is very exciting, and some of these switches to turn them off in normal tissue by recognizing class 1, for example, but then when class 1 is missing, able to recognize the antigen target. I'm excited about that, and if I can also echo Pedro's comment about the tumor microenvironment, all of these technologies that can now not just look at a lot of markers, more markers is always better, you learn more, but the informatics ability to make the most out of that, and the ability to think about the nearest neighbors, who's actually next to the tumor, who's talking to who, and how does that really impact the therapeutic outcome. So that's another, I think, exciting area that we're just scratching the surface of, that's going to give us some great insights.
Thanks, Lisa. That's a really good point about the informatics. I remember when I was a fellow doing those chromium release assays, it was really easy. I would just put the data in my own little spreadsheet. I know the answer as it was coming off the machine. But now we have so much information. One CyTOF experiment, a single cell RNA-seq, really gives so much information. The spatial area that you were talking about, also very important. How are we going to deal with all of that information across immunotherapy? Are we going to see some papers coming in through JITC of artificial intelligence interpreting our results for us because they're so complicated? What do you think the future holds for all this information that we're now getting in our immunotherapy research studies?
You're right, Patrick. There is a huge flood of data from all kinds of approaches. And at the journal, we are very excited about working together with the society to try to put together a series on data science, big data processing, machine learning, artificial intelligence, from the webinars that are going to be taking place probably starting this year, sometime this year.
Yeah, that's so important. I can't hire anyone from my lab now that can't code in R because they're not going to be able to analyze their own data. So that's a great thing that the society is now having task forces and articles to, because it's not intuitively obvious. There are mistakes that can be made, and to share best practices on the data analysis is going to be very important.
Yeah, so it's really wonderful how JITC is going, and the skyrocketing impact factor that Andrew talked about. But then, as James said, it's getting more competitive to get papers in. It's what would you guys give as advice for especially young authors and investigators out there? How do they get their papers in this increasingly competitive journal, JITC?
I think something that works very well, in my view, is to contact us have pre-submission inquiries, and of course, as was mentioned before, translational research papers are great, and so those papers with a richness of translational research ingrained in exciting clinical trials. We encourage very much our young authors to go for them and to really put excellent science, and we will be very happy to consider those pieces of good results.
Lisa, you do a lot of mentorship for young people. What would you give them advice to get a paper into the really competitive journal, JITC?
I think about human relevance. If there's human relevance, clinical relevance, that's number one, because that's the audience. That's the audience of translational science and physician scientists who are reading the journal. And then, I think about, where we are technologically, and everyone doesn't have to be on the bleeding edge, obviously, of every new technology, and not just technology for technology's sake, but, to have, to be thinking about things in an up-to-date, technologically sophisticated way. Those are things that are, going to be exciting. They're going to have a lot of data, a lot of interesting data. And so those are things I look for. And then, of course, don't annoy your reviewers. Don't proofread. And then, that's another important, simple thing everyone can.
James, what do you tell your young mentees? We're looking at JITC.
Yeah, so I say, first of all, we got to look for what we're looking for as editors. We're looking for exciting science. We're looking for first in class, for first in human. We're looking for things that really can set the tone, set the pace, or some really unexpected finding. And I would agree with Pedro that we really do like those pre-submission inquiries. They can help expedite things. They can help get things going more quickly. I also want to just give a shout out to Andrea and the team and Pedro. That we've really gotten to the point where we have set some very aggressive turnaround times for reviews, and I think we've done a great job there. So I think it's a great place to submit really high quality science.
Andrea, how do you do that in terms of getting those rapid turnaround times, which has really been quite impressive? Because when an author sends in their paper, they really want to get an answer back pretty quickly. And I know that all the reviewers that you have are very busy. They all have full-time day jobs and an awfully competitive academic environment. So how do you engage them and make sure they get those turnarounds going?
Yeah, well, it's not easy. So it's obviously, as you mentioned, everyone's so busy. And we especially felt that with the pandemic, as people became busier in their communities and their institutions, the time that they had to put in towards reviewing for journals became much more limited. And we felt that we really saw that across the publishing industry. Working with reviewers and engaging them can be a challenge. And so at the journal at JITC, we really do work to try to help motivate and provide opportunities for engaging and going beyond. And so with our reviewers, it's beyond just doing, an annual thank you to them. We've increased our opportunities to provide continuing medical education. So we do have CME credits available. We collaborate with Publons and other services that provide opportunities to acknowledge reviewer activity at journals through their own profiles. And even with SITC, we have worked with leadership to include reviewer opportunities or reviewer activity as a way to kind of help in your path towards leadership at the society and opportunities at SITC. And we look to that too at the journal. The reviewers, it's a benefit to be reviewing for JITC because we always go to the reviewer performance as our first source when we're considering opportunities for open editor positions. And so there are a lot of ways that we try to really help build an experience for the reviewers who do work with us. Even with as busy as everybody is, yes, we know we send a lot of emails to try to make sure we get reviews in on time. And we hate to have to do it, but the reviewers that we do have who do commit with us, we're very fortunate that they provide pretty fast turnaround times to really help get the author's information out to the public as soon as we can.
Wonderful. Well, Dr. Gully must have been really great on the turnaround times if to earn that deputy editor-in-chief title. Andrea. Andrea, while we on the subject, how we view materials has really changed throughout the years. I'm old enough to remember I used to go to the library at the NIH. I'm sure James has been to it, and we would go there with Xerox articles, or we look at the hard copy of the magazines on the rack. Now, no one goes to the library, it's all online. But then there's all of these other mechanisms. Jitsi has a wonderful Twitter site, where there's a number of tweetorials I've learned, and it really helps you in a very brief way keep up with all the information out there. Can you tell us a little bit about Jitsi and the initiatives that have this multimedia reach that can get out there and reach multiple countries throughout the world in multiple platforms?
Yeah, absolutely. Jitsi, we are blessed with a really great social media editor, Dr. Jason Luke, who oversees a social media team and really helps to engage researchers throughout the world who use Twitter. And with that, getting new articles out there to everybody, every single article that gets published in Jitsi gets that kind of recognition and beyond, and keeping people up to date, even at what's going on in other journals, what's going on in the field in general, just kind of creating a resource for our followers. So definitely the way that Twitter has evolved for Jitsi has been very helpful in extending the reach of our articles. And we're even now starting to trying out a pilot program with in China, just to go through kind of the WeChat platform. And so really being able to extend on those different platforms, again, gets material in front of people. And even within the journal itself, being able to use what's kind of up and coming in publishing trends and incorporate it into our articles, it just extends the reach further, such as, video abstracts. We just published our first video abstract this past year, and are really looking to take that much further in the coming months. And then even graphical abstracts, which we've been publishing for a couple of years. All of these types of things just really help to create another media form to be able to digest and easily take in that information that is being displayed in these articles that we publish.
That's great. That really, I really enjoyed those tutorials. And, the other thing I've seen is sometimes on the Jitsi site, you talk about articles that aren't in Jitsi that are really important to the field, which I really appreciate, that you're really going across different journals.
Yeah, we have our editors engaged each month. It's called Jitsi's Reading List. And so we ask a different editorial leader to identify 4 or 5 articles of interest to them that they read in other journals. And we share that on a regular basis. I think it's around the 3rd week of every month that we update that and include a mention in our digest. And it really does help to serve everyone, just provide a service to our readership, expanding beyond even what Jitsi publishes, but what our editors think and what interests them and what's advancing them forward.
Now I'd like to bring out a question to the group. Jitsi and immunotherapy has had a broad influence across many different sciences, including infectious diseases. The first RNA vaccines were really started in cancer vaccines. And I know, Dr. Romero, you have had such a long history with cancer vaccines as an expert there in Lausanne. Tell us about COVID, what Jitsi did during COVID. How did it change the practices and getting the journal going through keeping it going through COVID in such a robust way. And also, there's a really nice series of articles on COVID and the immune system. Tell us about that.
Well, the COVID times actually meant for us a hike in workload because we receive an increase in submissions. People all of a sudden had time to concentrate on writing their pending articles. And so this was very good for the journal. And indeed, we managed to publish a series that was guest edited by a couple of very dedicated and insightful fellows. So the one was Paolo Ascierto from Naples, and the other one, Jason Goldman, actually from infectious diseases in Seattle, in Seattle University, Washington University, sorry. And so this series actually tried to cover different aspects of the COVID pandemic and immuno-oncology, the implications for cancer immunotherapy, the interactions, and as you mentioned, the vaccines, in this case, the contrast between prophylactic vaccination with things like mRNA as delivery vehicle and therapeutic cancer vaccination using the same, and that crystallized in a series of articles that are proving very popular. One, for instance, that comes to mind is actually a paper from an oncologist here in Lausanne, Solange Peters, with several others, experts in lung cancer, that talks about severity of COVID-19 in lung cancer, evidence and challenges. And there are other things like immune profiling of COVID-19, led by Holden Mecker, and inflammatory cytokines in immuno-oncology and in COVID-19. So it's, I think it's a very exciting series, and it really is unique in bridging or integrating our new knowledge in COVID-19 and immuno-oncology. How is COVID-19 presentation, management in cancer patients?
That's great. And there's so much interactions between cancer vaccines and tumor immunology and infectious disease vaccines, the basic science of immunology and translation. It's really great that JITC was helped to bring all that together. And it was wonderful seeing everyone at the annual meeting in November in person, where we had Ugur Sahin and Aslam Turechi, who were SITC members, cancer immunologists, who really helped to develop that first RNA vaccine, the BioNTech Pfizer vaccine for COVID, and they received the Medal of Honor there. So it really shows the tight interaction between infectious diseases and tumor immunology. Well, this has been just a tremendous discussion, you guys, a celebration of 10 years of JITC. Anything else you guys would like to add about our 10-year anniversary?
Maybe I like to take the opportunity to stress that this has been a collective effort that has been tremendously supported, always very generously by you, the leadership in the Society for Immunotherapy of Cancer. And I think we have a bright future ahead of us, continuing this adventure of publishing open access, excellent tumor immunology and cancer immunotherapy science.
Wonderful. Well, what a celebration. 10 years of JITC. Dr. Pedro Romero, James Goley, Lisa Butterfield, Andrea Kuntz, thanks so much for being with us today.
Thank you, Pat.
Thank you.
A lot of fun.